The first ever vaccine target for trypanosomes, a family of parasites that cause devastating disease in animals and humans, has been discovered by scientists at the Wellcome Sanger Institute. By targeting a protein on the cell surface of the parasite Trypanosoma vivax, researchers were able to confer long-lasting protection against animal African trypanosomiasis, AAT, infection in mice. The study, published today, the 26th of May 2021, in Nature, is the first successful attempt to induce apparently sterile immunity against a trypanosome parasite. A vaccine was long thought impossible due to the sophisticated ability of the parasites to evade the host immune system. As well as a strong vaccine target for AAT. The findings raise the possibility of identifying vaccine targets for trypanosome species that cause the deadly human infection sleeping sickness and Chagas disease. Animal African trypanosomiasis, AAT, is a disease affecting livestock in Africa and, more recently, South America. It is caused by several species of Trypanosoma parasite, which are transmitted by tsetse flies, causing animals to suffer from fever, weakness, lethargy and anemia. The resulting weight loss, low fertility and reduced milk yields have a huge economic impact on the people who depend on these animals. The disease has been said to lie at the heart of poverty in Africa. In humans, a disease called sleeping sickness is caused by infection with another trypanosome species, Trypanosoma brucei. Although control efforts have reduced the number of infections each year considerably, 65 million people remain at risk. In South America, the potentially life-threatening infection Chagas disease is caused by Trypanosoma cruzi and affects at least 6 million people living in endemic areas too. All trypanosome species have developed sophisticated anti-immune mechanisms that allow the parasites to thrive in their host. For example, African trypanosomes display a protein on their surface that constantly changes and prevents host antibodies from recognizing the pathogen. Until now, it was thought impossible to vaccinate against trypanosome infection for this reason. In this study, scientists at the Wellcome Sanger Institute analyzed the genome of T. vivax to identify 60 cell surface proteins that could be viable vaccine targets. Each protein was produced using mammalian cell lines and then used to vaccinate mice to determine if the host immune system had been instructed to identify and destroy the T. vivax parasite. One cell surface protein, named invariant flagellum antigen from T. vivax, IFX, was observed to confer immunity against infection in almost all vaccinated mice for at least 170 days after experimental challenge with T. vivax parasites. Thanks for watching. Please subscribe my channel for more updates.